What I want to do is cover microbes and pathogens and microbial indicators. And if we have a little bit of time, I know your homework handles toxicity. If we have a little bit of time, we'll look at aquatic toxicity today, although I think I'd like to sort of cover it a little bit more thoroughly later on. The agenda over here is to learn and not to sort of meet all sorts of goals and time slots. So hopefully learning is taking place. Let's go ahead and talk about microbial indicators. The one thing, the reading in your book was this guidance to implementing the Ecoli and Enterococci standard, the EPA guidance, and I think I gave you like from page 1 to 3 and 5 to 10. You need to read it and understand it and make sure you understand all the charts and the graphs and stuff like that. And I'm going to explain that to you in a minute. But essentially what we want to do is, why do we care about bacteria? I mean, what's the big deal here? And not just bacteria, but microbes like Pfiesteria and Clostridium and all of those kinds of things. Giardia. Anyone had Giardia before? Anyone know anyone who's, yeah, it's pretty bad stuff. Yeah, so all of those Campylobacteria. Why do we care about that stuff?
Disease.
It's a public health issue, and the majority of the bacteria we encounter in nature are pretty much harmless. You know, they come in and they go out and nothing happens. But there are certain bacteria in nature that are what we call pathogens. In other words, they react with our bodies. So, for instance, bacteria, we might find, for instance, most of the E. coli that we have in our systems, but there's a little strain called O57, H157. Is that? Let me just make sure I have the. Oh, I don't even have it here. But there are certain strains of E. coli that are incredibly toxic or pathogenic to us. So, for instance, E. coli, the Shigella virus, Campylobacteria, these are viruses that give us, we sometimes get viruses that give us diarrhea. And so, you know, you eat something, man, and the next thing you are puking and going to the bathroom and, you know, you're there the whole night. But there are other bacteria that has anyone had dysentery before? What is dysentery? What's it? And we're getting kind of nasty over here, but what's the difference between diarrhea and dysentery? Dysentery is where you get so freaking sick that these bacteria basically attack the lining of your alimentary canal. And so instead of pooping water and bits and pieces of organic matter or organic waste, you're pooping blood. And so things like the Jack in the Box outbreak in the West, and then Taco Bell, that's not people, oh, I'm sick, I've got to go to the bathroom, oh my goodness, I was in the bathroom the whole night. This is people losing blood through their alimentary system. Okay? And so that's when it gets really, really bad. Viruses. I mean, we're in a generation, in my generation, I saw maybe 5 or 6 or 7 kids in my entire youth that were walking around in braces. But polio was a huge thing. You go to Africa, polio is a huge thing. So Rotary has tried to stamp out polio, and generally they've done a good job. March of Dimes, 40 years ago? That was their first thing. But polio is a waterborne virus that will attack, and mainly it attacked children because that's the kids would go swim in the mud holes and stuff like that, and they'd come home and they'd get sick and they couldn't breathe, and that was a huge deal. So these are protozoans. Giardia, I think it's also called giardiasis, is also called hiker's disease, and they say that is probably some of the most evil smelling poop when you go to the bathroom, so you don't want that. Cryptosporidium, there's a little town in Ireland that was infected by Cryptosporidium, the tiniest oocysts, so of basically a little protozoan. And man, those oocysts get through any kind of water system. You can treat it with chlorine and filter it and stuff like that. Those oocysts get in your system, it's bad. It's bad. And when you've got an entire town that's infected with Cryptosporidium or crypto, it's really bad. So those are pathogens. Most bacteria, again, or most microbes are not pathogenic. Viruses, and that's passed through our system. So we have something called indicator bacteria. And why would we do that? Why would we have indicator bacteria in the first place? Just for fun, to keep the scientists sort of on a stipend. Okay, typically a good indicator bacteria is a bacteria that has a similar life cycle and similar, basically a similar life cycle to any of the other pathogenic bacteria you might find. And so one of the ones that we find in this country, one of them is, well, let me have a look. We've got 3 indicator bacteria that we use most of all. One is the total coliform group of bacteria. One would be the fecal coliforms. Another would be the Escherichia coli species. And then finally, enterococci, or I think it's they pronounce it enterococci. I don't like pronouncing it like that, but I think that's the correct pronunciation. So those are 4 indicator bacteria. Why are they good? It's because we find them in the guts of warm-blooded creatures. And so typically, if that happens, they're found in the gut. So when you excrete that through feces, you're going to find them in whatever receiving water body there is, if, for example, those feces don't go through some kind of treatment system. The indicator bacteria are numerous enough that they'll always be present, and then we can basically find a correlation between the concentration of those indicator bacteria and maybe illness. And that's what this document was about. There was an epidemiological study. Okay, so let's very quickly look at that epidemiological study. Essentially, what they found was, for instance, fecal coliform and total coliform, when you started to plot the dose response, curve between fecal coliform and even worse, total coliform and illness, you had data that kind of looked like this. And you kind of drew your line, and basically it didn't, no, sorry, it's the other way. Sorry. Illness on the x-axis and fecal coliform. So kind of they started looking and they got some data out here, and they said, well, fecal coliform and total coliform, there's no, you vary your concentrations of fecal coliform, and even though these things come from the guts of warm-blooded creatures, there's no variation in illness. So, that's not really good. So in, I think about the 1960s, there was a guy, and I can't remember his name, an Italian dude that started an epidemiological study, because really what you want is you want a dose and response kind of thing. Your dose would be bacteria, and your response would be illness, right? So really what you'd want is kind of a nice dose-response curve to say, okay, if my bacteria is at this concentration, then my illness rate per 1,000 is that, okay? So that's whatever this guy and his cohorts went ahead and did. And so what they did was they went to 7 lakes, and they also went to seaside resorts. And over a summer, they basically went to people who went down there and swam, and they would go to them and they'd say, well, did you swim today? Yes, I did. Did you submerge your head underwater? No. Okay, well, we won't talk to you. Okay. Did you go and swim today? Did you submerge? Yep, did. Okay, can we get your name and address, and can we call you in 3 days' time? Yeah, sure. And so they did that to about 3,000 swimmers over the period of that summer in each of those 7 locations. And then they would call the swimmers up 3 days later and say, hey, just calling you. Remember, we were out there. Now, of course, they had banks and banks of people. Just calling you up and saying, you're doing okay. How are you feeling? And the guy would say, oh, actually, I had diarrhea, or I've got a cold and I'm coughing and I don't know what's going on. Okay. And so they would say, okay, was their illness related to this waterborne disease or not? And typically they would have very strict criteria. And what they found was, and you can actually look at the graph on page I think it's page 8 of this piece of paper. And what you actually found was this, well, I drew the line first, was a very good plot. Each one of these represented a particular location, the beach, and the average illness rate per 1,000 people. Okay, and this was my E. coli population. And what they found was that E. coli, the concentration of E. coli, was a very, very good predictor of illness in terms of illnesses per 1,000. And they also found that in salt water, enterococci or enterococci were good both in fresh water and salt water, good predictor of illness versus concentration. E. coli wasn't a great predictor of illness in salt water. Okay, so what I just wanted to sort of point out to you, that's kind of the epidemiological studies, and we base so many of our regulations on like this one epidemiological study that happened years and years and years ago. So we don't know if the concentration is, say, 200 CFUs per 100 mils down on the Saluda, that if we had to have 3,000 swimmers go in there, that we'd have the same illness rate. We just don't know. But we're just using that epidemiological data. So people don't necessarily want to pay for that kind of data. But that was kind of the gold standard study that they did way back then. So that's kind of what I wanted to convey to you. An indicator bacteria then is, in it and of itself, it's not pathogenic. So E. coli, unless there are certain strains, E. coli in and of itself is not pathogenic. Neither are fecal coliforms, neither are total coliforms, neither are enterococci. But you will find them in the guts of warm-blooded creatures. And their life cycle, especially after they exit the body, they're in a secondary environment and they rapidly decay. Typically, if you have 1,000 E. coli per 100 mils today, you'll have 100 tomorrow, you'll have 10 the next day, you'll have 1 the following day. So there's that exponential decay of E. coli simply because they're out of their natural environment. Okay? So let me just talk very briefly and this will probably be an exam question here. Okay, so forewarned is forearmed. We have these 4 indicator bacteria, and what kind of one of my pet peeves is that when you start talking to lay people and people in the newspaper, goodness gracious, don't get me started about people sounding the alarm in the newspaper, is people in the newspapers, lay people, kind of get E. coli, fecal coliform, and total coliform. They use all of those terms interchangeably. And what I want to do with you right now for the next few minutes is be very, very specific about those 3 bacteria. Enterococci is in a group all on its own. So let's first start with the universe of total coliform bacteria. Okay? Total coliform bacteria is made up of something like about 100 species. Or is it? No. It's been around, 18 species. It's been around for about 100 years. But about 18 species have been found in this total coliform bacteria group. There are many of those bacterial species that you'd find in our bodies, but there are also many of those species that you'll find in nature, like Klebsiella, you'll find in plants and stuff like that, and have nothing to do with the bodies of plants and animals. So with all of those varieties and stuff, you can imagine that it might not be the greatest indicator. Now, I'm not really going to go and jump into water that has a high total coliform bacterial count. But it might be, I think, there are certain things like, I can't remember, effluent from paper pulp mills or something that might have a high count of Klebsiella. I can't remember what it was. But not all of those things have an animal gut origin. So typically they're not going to really be harmful to us. So that's this whole universe. Operationally, these things will break down, will oxidize. Let me just quickly have a look. They will ferment lactose. At 35 degrees C. Okay? And some people have it at 28 degrees C. I don't know, Europeans sometimes do it differently. So they will ferment lactose at 35 degrees C. So they're all different kinds of species. They're also, I think, gram-positive. Are they? They're aerobes as well. So they usually live in aerobic environments or facultative aerobes. In other words, if you put them in anaerobic environments, some of them will still survive and they'll change their metabolism. And I think, I can't remember if they're gram-positive or not. I don't care whether they are or not. But they are aerobes or facultative anaerobes that ferment lactose at 35 degrees C. There are about 80 species, 19 genera. And this is total coliform, or what we now know as coliform bacteria. It's an incredible general group. As an indicator bacteria, it's kind of lost its use. Okay. Now, I need to look at that. In that subset, I'm going to say 28 degrees. Okay. In that subset, we have something called, 35. Right, reset. In that subset of total coliform bacteria, we have a group called Fecal Coliforms. They are thermotrophic. So what's thermotrophic mean? What does it mean to be thermotrophic? Thermo means? Heat. They love heat. Man, they just love it. Okay? That thermotrophic means that they live and grow fat at high temperatures. In fact, if you had to take a sample of all of these and incubate it at 35 degrees, you'd get all of these fecal coliforms. But if you had to do exactly the same experiment and incubate at 44.5, what would happen is you would kill a whole bunch of these guys, and you would end up with the fecal coliforms. Now, the logic behind that is that thermotrophic bacteria are typically going to be more associated with the guts of warm-blooded animals and less associated with trees and roots and plants and that kind of thing. Makes sense? Okay, so these are, I don't know how many species and genera there are in the fecal coliform bacteria, but they are thermotrophic. Now, E. coli or Escherichia coli actually makes up around about, let me do this. E. coli is a single species. Escherichia coli is a single species that's about 88%, anywhere from 80 to 95% of fecal, and usually spelled fecal coliform with small letters. Escherichia coli would be italics and capitalized. Okay. E. coli is called the fecal coliform bacteria because typically about 80 to 90% of them is actually this species E. coli. So this one is a very, very specific species, whereas these are not quite, and of course this is very, very general. So you can see why the epidemiological studies would say that as an indicator, the total coliform bacteria is pretty useless. I'm not quite sure why the fecal coliform bacteria were not a better indicator than those data suggested, and why E. coli was so much better. Because if E. coli make up 88% of the total or fecal coliforms, it doesn't make sense to me. But that's another story. But I want you to understand there's a difference between total coliforms, fecal coliforms, and E. coli. Just because they have the word C-O-L in them, based on the colons of warm-blooded creatures, you need to understand we don't use them interchangeably. And if you use them interchangeably in classes or in homework, I will probably come and shoot you in your house. Well, that's a threat, I mean. I'll know where you live. Okay, so it's real important because, one of my things about water quality is that we have to be careful of where we get our science. And when people in the newspaper say, thousands of E. coli's, thrown into, and it's like such blather and baloney of these guys. They have no idea what they're talking about. And when they're sort of stating it and trying to educate the public, oh my goodness, we've got E. coli in the water. Yeah, we have E. coli, but the E. coli is not the thing that makes you sick. It's an indicator of other things. And we have to look at it as a probabilistic thing. I'm not saying we need to pour E. coli into the water, but we need to be considered and understand what those implications are, because total coliform bacteria in the water is very, very different from E. coli's. I mean, I've gone out and got less than 20 E. coli bacteria in the water, but more than 2,000 total coliform bacteria in the water. So it's a huge difference, and we need to, as scientists or as people who talk to those scientists, we need to distinguish between that and kind of tease that stuff out. I think with quite a few of you guys, I've kind of been nitpicky about the language you use. We need to make sure, don't interchange a word like total coliforms or coliform bacteria for E. coli. Okay, enterococci are pretty much in their own ballpark, and so they don't really come into that equation. Or they're a completely different set. They don't ferment lactose or anything like that. Last word, and then I'm going to let you go. Do you notice they use the geometric mean instead of the average? For some of those regulations. So why is that? Why do we use geometric means instead of, why don't they say, they use averages for DO, but why don't they use it for fecal coliform bacteria? If you have a look at the frequency distribution, that's your frequency and that's your concentration of bacteria. What happens is that over time, you find something like, so these are, okay, I go out every day and I measure that. And over time, these are the number of samples I'm going to find in different things. I'm going to find samples here, and, sometimes I'm going to find samples way up there. And you kind of get this kind of what approximates a log normal distribution.
What were the x's? I'm sorry.
Oh, I'm sorry. This is a concentration, and this is the frequency with which I found those samples in here. So you've got a frequency distribution that, what does a normal frequency distribution look like?
It's like a normal curve.
Like a bell curve, right? I get one sample here, and more samples here, and more samples here, and more samples here. You'll find that with, for instance, dissolved oxygen, and I've noticed even with phosphorus in the water, you've got a nice normal bell curve. So if I take an average of these concentrations, where's my average going to be?
Top of the bell curve.
It's going to be the same as my mode, average mode, and it's going to be the same as my median, right? But I do that over here, what's going to happen with my average? What's going to really influence my average here? Is my average going to be sitting up here?
It's going to be, the outliers are going to pull it to the right.
Those outliers are going to make my average sit here. So is that an unbiased estimator of this population? In this case, it's not an unbiased estimator. And in fact, what you find is the geometric mean, basically you take the logs of each, and then you add the logs up, and then you divide the logs by, you average the logs, and then you take the antilog of the average of the logs, and you'll come up with that geometric mean. That geometric mean comes out right here, and in fact, it looks very similar to your median as well. And in fact, very similar to your mode. Okay? It's just that the mode is difficult to calculate. That becomes a much better unbiased estimator of the population of bacteria in your water. Remember, we can only take samples, so we can make inferences about that population. We don't know what the actual population is. So that's just kind of why do we use geometric means for bacterial populations? It's because they don't have that nice normal bell curve like we find in other things. In fact, a lot of things in nature have this kind of log normal curve, what we would call a log normal curve. All right, I've taken up more time than you had paid for, so I'm going to dismiss you. Readings, I'll have a look at the readings, but next week we'll continue to look at toxicity. But in fact, next week we'll start looking at basic hydrological concepts. So go ahead and have a look at those readings as well. We're going to have a look at a little bit of sort of the big picture hydrology in our water quality. Okay? All right.